welcome everybody to our October 17th mini seminar series. As usual, we're going to go through a couple of housekeeping items before we get to the seminar. This month happens to be a 4-year anniversary of this working group that was started initially on October 22nd in 2020. And our first regular seminar was in November of 2020. So we've had, I think, over 200 seminars that are available on our YouTube channel. And also, as a reminder, this webinar is being recorded and also live streamed on YouTube as we speak. We have a wiki page with all the previous seminars, but unfortunately it is down right now, and Jim Sluka is working with NIH admins to get it back up. So hopefully it'll be working soon here. As always, James and I are always happy to take any questions, suggestions. We also wouldn't be able to run this group without Jim Sluka and Lorenzo, who help us keep the website going, invite new speakers, and sort of manage different aspects of running this group. We do have several social network and information pages ranging from Slack, X, wiki pages, LinkedIn, and YouTube. If you have any announcements, we're happy to share them there. And also feel free to follow these resources for new updated information. Twitter or X is also one of the more actively used tools. So again, if you have any posts that you would like us to share, please let us know. Also feel free to engage with the channel and the posts that happen there. We also have a reach to a lot of the community members, and so if you'd like to advertise your publications that you published in the area of multiscale modeling and viral pandemics or digital twins, please you can email Jim Sluka, and we'd be happy to post it on the website. We also on the Global Alliance for Immune Prediction and Intervention, or GLIMPRINT.org website, we can also now post directly job listings, webinar announcements, seminars, funding opportunities, and so on and so forth. So you can go either to this link and submit your announcement, or again email Jim, and we thank Jim for he will post it for you on the website. We have several available positions. Again, you can post them directly on the website, and if you email us, we'll also advertise them here during our webinar series. We have a couple of positions available, faculty positions at the University of Florida, working with Reinhard Labenbacher. If you're interested, you can check out the positions on this link, and you can also email Reinhard directly for more information. At the University of Nebraska, in our Digital Twin Innovation Hub, our group is currently looking for several staff scientist positions in multiscale modeling and agent-based modeling. We're developing a immune digital twin infrastructure from the models to the software, user experience, and all of that stuff. So if you're interested, or you know of somebody who may be interested, please feel free to email me directly at this email address, and I'd be happy to share more information and discuss. Jason Shoemaker is also recruiting students and postdocs for his recent grant on computational modeling, hormones, and influenza infection. So again, if if you are a student or a prospective student, or you have any students or postdocs who may be interested, please email Jason at this email address. And he's at the University of Pittsburgh. Several funding opportunities are also currently active. One of them is NSF Pathways to Enable Open Source Ecosystems. So if you're developing, not necessarily modeling, but if you're developing software, open source software, and you're building a community around the software, this may be a good opportunity. There will be exactly a year ago, exactly in a year, in 2025, there will be a workshop on virus dynamics in France. Here is a link that you can learn more information, and also for registration. There will be, at the end of this month, there will be a workshop at the NIH on AI in precision medicine and diabetes and other chronic diseases. I am not sure if there is still a way to register, but I would imagine so. These workshops are relatively flexible, but this is happening in the next couple of weeks. Another funding opportunity from the NIH that focuses on systems modeling of infection and immunity across biological states. This is applicable to, this NOSI applies to R21s, to the R21 and R01 funding mechanisms. And these are, excuse me, this call is open anytime now, basically after October 5th, during the standard NIH funding deadlines. Are there any other announcements that anybody would like to make? Again, funding, meetings, talks. No, okay. Tomorrow we will have our steering committee at 11 o'clock Eastern time. If you haven't been invited, and it is something that you would like to join and contribute to the organization of this group, please feel free to email me, and we'd be happy to include you in the mailing list and have you join the steering committee. We have a full lineup of presentations. Thank you for those who volunteered to present, Lorenzo for inviting all these great speakers. We have the next available date for future presentations is after January 19th. I'm sorry, January 9th. So if you're interested in giving a talk, or you have suggestions for somebody for us to invite, please feel free to either email us directly or go to this URL, and you can suggest speakers that way. So that concludes our housekeeping items. It is my pleasure to welcome Vladana Vukojevic. Is that right? Am I pronouncing that right?
Yes, perfect.
Okay. To give our 4-year anniversary webinar. Thank you.
So thank you, Tom, for the introduction, and thank you for joining this webinar. It is a privilege to be able to present our work to you, and I'm, of course, most grateful for this opportunity to the organizers of this seminar series. I am a newcomer to the group, so I would like just to briefly introduce myself. As you have heard, my name is Vladana Vukojevic. I am and let me see now, this is not running. Yes, I'm an associate professor of biochemistry at the Medical University Karolinska Institute in Stockholm, Sweden. And my research focuses on cellular and molecular mechanisms that underlie the development of alcohol use disorders. And we are in particular focusing on the role of the opioid system in the development and management of alcohol use disorders. And the reason why we are focusing on the opioid system is very simple. While alcohol use disorder is a common and rather widespread disease, there is actually only a handful of medications which are available to motivated individuals for treatment of alcohol use disorders. And one of these treatments is the opioid receptor antagonist, naltrexone. Now, pharmacotherapy of naltrexone is not fully understood, and we would like to focus on the molecular mechanisms, but also ways how one can improve it and make it more effective as a treatment. In order to understand how ethanol or alcohol is perturbing the properties of the opioid receptors, we are looking at live cells, and we are using advanced fluorescence microscopy-based techniques and time-resolved nanospectroscopy techniques with single molecule sensitivity, such as fluorescence correlation spectroscopy, in order to characterize quantitatively these effects. I will, however, not talk about this part of our work. I will rather focus on the other partner in the opioid system, and these are endogenous peptide ligands of the opioid receptors. In particular, I would like to focus on beta endorphin, which is endogenous peptide ligand of the mu opioid receptor. And in the central nervous system, it is produced as part of a dynamic network that we have in our organisms, which is called the hypothalamic-Pituitary-Adrenal axis. Now, as my title has indicated, my talk will actually have two parts. I would like to begin by presenting you a method which has been developed some 30 years ago, and it is particularly dedicated to take advantage of the oscillatory dynamic around a supercritical Hopf bifurcation in order to provide reliable quantitative measures that are characteristic for an oscillatory chemical reaction or a biochemical system that has inherent nonlinearity and is run under conditions close to a supercritical Hopf bifurcation. And this method is not only experimentally really valuable, but it can also be used to test model predictions. And I will talk a little bit about that. And in the second part of my talk, I would like actually to share with you the insights that we have gained by applying this method when we modeled neurochemical transformations that underlie the hypothalamic-Pituitary-Adrenal axis. Now, as I have mentioned, quenching of small amplitude limit cycle oscillations or quenching analysis is a method that has been developed some 30 years ago. And the people who have developed it are researchers at University of Copenhagen, the chemistry department, Preben Gros-Horesen and Finn Höne. And I was very fortunate actually to join them at the time when their second paper on the general theory of this method has been published. So my task was to assess the generality of this method. So my task was to set up another chemical oscillatory reaction, which is called the Briggs-Rauscher reaction, which belongs to a group of oxyhalogen chemical reactions, and it's popularly known as the iodine clock system because it exhibits changes in color from colorless to yellow to dark blue based on the concentration of intermediates and most importantly iodine and iodide. Now, when this reaction is conducted in what is called a closed reactor, or in a laboratory beaker where we just put in the reactants together, then the system undergoes a number of transient states. And as it is winding out, it is approaching, it is going to monotonous evolution and approaching the thermodynamic equilibrium monotonously. If the reaction, however, is conducted in what is called a continuously fed well-steered tank reactor, or a CSTR, then one can maintain a particular dynamic state which is stable under the conditions, under the examined conditions, for quite a long time, or indefinitely actually. So what is a CSTR? It is a reaction vessel which is typically thermostated, where one is flowing in the reactants and keeping the volume of the reaction mixture constant by removing the excess of the volume that is being formed. If one is flowing very, very fast, the reactants, then one is just simply mixing them, not allowing time for chemical reactions to take place efficiently. But if one is not doing that, if one is doing this slower, then reactions which can exhibit oscillatory dynamic behavior can be studied reasonably well. And here you can see, for example, periodic oscillations, which we have recorded in a continuously fed well-steered tank reactor. Now, I have mentioned that we need to study the system in the vicinity of Hopf bifurcation. And what we have typically done is we were changing the flow of the reactants, and when the flows are too low in this particular case, then the system is exhibiting monotonous evolution, or a steady state, a stationary steady state. And if this control parameter is then being changed or increased in this case, periodic oscillations are emerging. If we then decrease the control parameter, then the system goes back to the stationary state. And in the vicinity of a supercritical Hopf bifurcation, we can actually find the bifurcation point exactly by plotting the squared amplitude of the oscillations as a function of the control parameter. And here you can see for this particular example where the bifurcation point is being located. Now, why is it important that we are close to a supercritical Hopf bifurcation? The reason for that is because we can then actually look at the state around the stationary state. And if we now go from the time series, which I have shown here in the experiment, to presenting what is actually happening in a concentration phase space, you can see that a supercritical Hopf bifurcation can be recognized by actually the system being in a stationary state or exhibiting stationary state dynamics, which is then being lost. Typically, in the cases that we have studied, I have never encountered another case. A transition is coupled also with a saddle focus here in the center, and then a limit cycle oscillation is presenting itself. So now we can linearize around this stationary state, the system, and examine it. So what is a quenching perturbation or a quenching of small limit cycle oscillations? In quenching of small limit cycle oscillations, what we are doing is we are adding a chemical species, and we are trying by trial and error to find a phase angle and the amount which we need to add in order to be able to shift the system from the limit cycle to the stable manifold. When we are successful in this effort, we can see that the oscillations can be temporarily stopped, and then the system is winding around this manifold going back to the limit cycle, as you can see here. If we are now moving along this limit cycle and using the same amount but at different phase angles, then we will see a phase shift. Or if we are perturbing the system at the position which is opposite to the position here, then we can see an increase in the amplitude because the system is moving away from the stable manifold. And as you can see here, so the point of this experiment was actually to demonstrate that there is a unique value for each species, a unique concentration or amount of that species, and a unique phase angle on the limit cycle oscillation that can quench the oscillations. So by performing quenching with different chemical species that exist in the Briggs-Rauscher reaction, we could acquire for these reaction species which are present in the reaction mixture the quenching concentrations and their phase angles. The phase angles are always expressed with respect to a reference point. So typically in these measurements, we were using potentiometric recording by iodide selective electrodes. And quenching of small limit cycle oscillations can also be performed by adding a species that is not originally present in the reaction mixture. This is typically done when we want to actually remove a species, and we are adding a species that is reacting with that species and removing it. Quenching by addition here and quenching by removal are actually or should be identical if the chemical reaction, the removing reaction is sufficiently fast. Now, the first surprise when we wanted to demonstrate actually this theoretical expectation in a system that is run at pH, at low pH, so about 2, we were very surprised to see that quenching of oscillations by adding hydroxide ions is not actually equivalent to removing protons from the reaction mixture, even though the reaction between protons and hydroxyl ions is actually really very fast. And this was very surprising, and I have performed very many experiments in order to make sure that this is not just an error, but I can later on explain you that we were able actually to numerically emulate this result. What you can then also do in quenching of small limit cycle oscillations is you can perform quenching by dilution. In this particular system, we could perform dilution by water, but we can also choose to have reaction species which are present at very high concentrations to not to change their concentration. So we were actually diluting while keeping constant the concentration of the acidity, manganose ions, or keeping their both constant during the dilution experiment. And dilution is performed in order to be able to acquire information about the overall concentration of the reactants in the system. So now a very straightforward and simple way is to compare how the predictions of a model are actually agreeing with experimentally measured values. And at the time when I was working in this field, there were two models for Briggs-Rauscher reaction that have been well established, and these were proposed by Furrow and Noyes and De Kepper and Epstein. And what we could see in our from our experiments is that the agreement between model predictions and real experiments is good for some, reasonably good for some species, but not very good for all species. And this immediately was an indication that we understand to a reasonably good degree, actually, the transformations in the iodine chemistry, but we don't understand really very well what is happening with malonic acid or with manganose ions that are also present in the reaction mixture as catalysts. So to see when to then further advance the mathematical modeling, I have then extended actually the core chemical part by adding reactions that include malonic acid, and in particular we wanted here to emulate the behavior of hydroxyl ions to simulate this experiment. And I think this was rather fortunate that we could actually achieve reasonably good results. So then we set off to, of course, emulated by numerical simulations, the perturbation sequences. And here you can see that we also see a very slow recovery of the system after quenching, as we have observed in real experiments. We could also then go to conditions which are far away from the supercritical Hopf bifurcation and test whether the model can also emulate complex dynamical behavior under these conditions. And we were really very happy to see that we could actually, in numerical simulations, see bistability. So here, depending on the initial conditions, we could see two different dynamics behaviors, even though all the parameters and are the same in both experiments. We could also imitate, at least formally, right, the shape of these very complex oscillations, and we could see sensitivity to bursting, but could not really have sustained bursts as they were observed in an experiment. For mathematicians in the audience, you have probably immediately recognized what is being done in quenching experiments. So in quenching experiments, we are actually probing the eigenvalues of the Jacobian matrix around a supercritical Hopf bifurcation. So with that, I would like to kind of conclude on the method. If you have any questions, please let me know. Otherwise, I could proceed to the next part, to modeling the hypothalamic-pituitary-adrenal axis and how we have used quenching experiments to probe the dynamics in this system. So if there are no questions, I can then go to just introducing briefly the HPA axis. So the HPA axis is a dynamical network that integrates and synchronizes the functions of the nervous and the endocrine systems. Main components of HPA axis are peptide and steroid hormones, and such as cortisol in humans or corticosterone in rodents, and aldosterone, which are steroid hormones. And the most relevant peptide hormones are the corticotropin-releasing hormone and the adrenocorticotropic hormone, CRH and ACTH. The HPA axis is a complex and highly dynamic neuroendocrine system, and it controls a number of physiological responses. In particular, it is part of our response to stress. The HPA axis is characterized by rhythmic dynamics. So we have circadian oscillations, and we are also having ultradian oscillations. The anatomical origin of the circadian oscillations is very well known. However, all efforts to identify the anatomical localization of ultradian oscillations have failed thus far. And this is why we believe actually that ultradian oscillations may spontaneously arise in the system as a consequence of the non-linear feedback loops in the system. In humans, I have mentioned we are having a circadian rhythmicity, so the cortisol levels, they are the lowest at the end of the day. This is our go-to sleep signal when cortisol is low, and then during the night, cortisol levels are gradually increasing and reaching in the morning a peak, which is a wake-up signal for us. And then during the day, cortisol levels are actually going down, but they are not going down monotonously, but rather exhibit ultradian oscillations, which have a period of some 10, 20 minutes, or even a little bit longer, depending on the individual. In rodents, the lead steroid hormone is corticosterone, and rodents are, of course, nocturnal animals, so their circadian rhythmicity looks different compared to humans. So their corticosterone levels, they are increasing actually during the night. The HPA axis is actually really important, and there is a number of diseases in which the dynamics of the HPA axis is perturbed. And it is also external sort of stimuli from the environment that can also affect the dynamics of the HPA axis. So what is really very well characterized and known is how these lead hormones are integrated in the HPA axis. So the corticotropin releasing hormone is a hormone that is secreted by the hypothalamus, and it is a signal which is then it is actually a precursor to pro-opiomelanocortin, which is then giving the beta endorphin that we will like to actually feedback loop to the HPA axis in the future, but we have not yet done this. Now, if we look at this part of the HPA axis between humans and rodents, this part is common. Where humans and rodents differentiate is actually in processing cholesterol and the biogenesis of steroid hormones. As a consequence of differences which are not at the biological or genetic level, but rather on the kinetics of different steps in humans and rodents, a consequence in humans is that cortisol is the leading steroid hormone, and it is corticosterone in the rodents. So there is a lot of underlying work to define actually mass action kinetic equations that are describing the neurochemical transformation in the HPA axis. And we have worked for a number of years. I will later on show you the reference where we started with a small model and used what is called stoichiometric network analysis in order to identify instability conditions in the model, so where it can exhibit oscillations. But mass action kinetic was used to explain for formulate the feedback loops between the leading hormone species. Using stoichiometric network analysis, we were able actually to find kinetic rate constancies where this set of chemical reactions is exhibiting oscillatory behavior. In order to be able to emulate the circadian rhythmicity, we did not really describe in details the circadian clock. Rather, we have introduced a periodic function that we have designed in such a way that it is actually imitating the asymmetry of the circadian rhythmicity in humans. And we have included this function as an external forcing function that is changing the concentration of the corticotropin-releasing hormone during the day. When this is done, then this oscillatory reaction, or oscillatory dynamic is observed, but now with different properties, and we see small amplitude oscillations during the night part of day, and then during daylight we see large ultradian oscillation. We could imitate, at least, without any additional perturbations, the ultradian and circadian rhythmicity in humans and in rodents using these mechanistic models that we have proposed. What is extremely important, and a lot of effort was given to this, is that the concentrations of all steroid and peptide hormones which are present in the model, they are actually physiologically relevant. So you can here see that there is a very good agreement between the reference values and the values which are predicted by the model. And this is true for humans and also for rodents. Now, having developed this model, we have then looked for the literature in order to see again how well this model can actually recapitulate the behavior of the real system. What are its predictive values? And what was one very important thing is that we could nicely see this inverted U response of HPA axis activity to glucocorticoids. And this is a physiologically very well known state, which is actually telling us that there is a range of concentrations when HPA axis dynamics is oscillating. So what is really good with our approach to explaining mechanistically what is happening in the HPA axis is that we can now add additional species which are not part of the HPA system and see how they are affecting the HPA axis dynamics. As we are interested in how ethanol is affecting HPA axis dynamics, we have then expanded the mathematical model that I have just shown to include ethanol. And from the literature, it is really very well known that ethanol is inducing an increase in the secretion of the corticotropin-releasing hormone, and it is well known that a metabolite of ethanol, acetaldehyde, is exerting an action on steroidogenesis in the HPA axis. So now, having expanded the model with equations that describe the ethanol effect on neurochemical transformations in the HPA axis, we could perform quenching or perturbation experiments to see how adding ethanol at very low concentrations, which are equivalent to having, let's say, a drink at happy hour after working. So this is equivalent of one's shot of strong liquor. You can immediately see that the HPA axis dynamics gets perturbed. And then, of course, if we are using a larger amount of ethanol, then the effect can be way better observed. So here you can see the effect of concentration of ethanol, which is increasing, the effect on cortisol is increasing as the concentration of ethanol is increasing. And you can see here this behavior. Now, what was very interesting, we have also looked at how long it takes for the HPA axis to recover after the perturbation. So after, let's say, having one drink or maybe having 5 drinks. And here you can nicely see that the model prediction agrees really reasonably well with what is known from everyday life, and that is that it takes a couple of days actually for the organism to recover after heavy drinking. What was also important and highly interesting is that we could demonstrate that the same amount of ethanol is not necessarily causing exactly the same effect on the HPA axis, simply because the HPA axis is dynamic, and depending on whether we are perturbing the system at the phase of ultradian oscillations where underlying physiology is increasing cortisol or decreasing cortisol, the effect of the same concentration of ethanol on cortisol levels will be different. And here you can nicely see that the same dose of ethanol introduced at slightly different times is actually inducing a very different response of HPA axis at the level of cortisol. And this was extremely important because in research groups that are working with animal experiments and trying to understand how ethanol perturbs HPA axis dynamics, they have reported opposite findings. And we could demonstrate that both groups, or all 3 actually groups, can be right, but depending on where the underlying HPA axis dynamics is, the response of the HPA axis to the same challenge of ethanol will be different. What was another thing that we could observe from modeling is that the response of HPA axis to concentrations of ethanol which are lower than 8 millimolar, so this is like 2 small glasses of drinks, is different. So here we could observe quenching of oscillations, but if we are using high ethanol challenges in the model higher than 8 millimolar, then we are always actually stimulating cortisol release. And this was again really very interesting because there is an NIH call, because experimentalists have actually observed that low doses of ethanol and high doses of ethanol exert different effects on the HPA axis dynamics. What you can also see here is that a challenge during the daytime and during the night, again, is not causing the same effect. And here, for example, we can see that a challenge which during the day is introducing measurable or changes that one can deal with is during the night actually really very perturbing. Another important consequence of modeling is that we could observe that ethanol-induced challenges are increasing actually the amplitude of ultradian oscillations during, let's say, the next day. And because the amplitude of ultradian oscillations is now increased, then stimulation with ethanol during the next day does not cause the same effect as observed during the first day. And again, this was really very nice because experimentalists have observed what they call a blunted HPA axis response to an ethanol challenge, consecutive ethanol challenge, which was not really easy to explain. Finally, we have tried to simulate numerically chronic ethanol use. And what was really interesting here is we could see that the HPA axis dynamics is undergoing a number of changes, and at concentrations which are very close to what is known from real life to be close to intoxication and coma induction, it is even flipping the dynamics of the circadian rhythmicity. So even though ethanol is by itself not affecting the circadian rhythm, and in our model we are having a periodic function, forcing function, we can observe an inversion, and instead of reducing during the day, cortisol is increasing during the day and falling during the night. And this may be the reason why chronic alcoholics actually experience an inversion in sleeping patterns. They typically cannot sleep during the night, but sleep during the day. What was also important for us to see is that the rhythmicity of HPA axis was totally destroyed at concentration of ethanol, which are 200 millimolars, which is the concentration for induction of coma that is known from real life. And now just briefly, our work, theoretical work, has attracted actually the attention of a colleague from the Mayo Clinic who was working on the development of a non-substance model for acute mania. And in these animal models, he is using what is called hypothalamic kindling or electrical stimulations of the hypothalamus in order to induce manic behavior. And our aim was actually to see how the HPA axis dynamics is perturbed under hypothalamic lateral kindling, and whether our mathematical model can emulate these experimental observations. So here you can see several things. Even though in the electrical stimulations are performed in pulses, which are modeled here, these electrical pulses are inducing corticotropin releasing hormone, you can see at the level of corticosterone that the change is smooth, even though the stimulus is pulsatile. And we could nicely see a reasonably good agreement between numerical simulations, here shown in black, with experimental measurements. Of course, this is not ideal, but it is simply because we could not have continuous sampling of corticosterone. So we are having 20-minute intervals at which we could sample corticosterone. And now we could, of course, make a number of predictions using the mathematical modeling in order to simulate experiments which would ethically not be approved because they would cause huge suffering in the animals. So I see that my time is slowly running out. What I hope that I managed to convey is that there is an experimental technique which is really easy to perform and to understand, which gives values which can be then easily compared with model predictions and can be used actually to evaluate how good the mechanism proposed by the model is reflecting the real system. And in the other part of my talk, I hope that I was able actually to convey why the same stimulus introduced at different parts of the day is not inducing the same response. In this particular case, cortisol response or a response of HPA axis to ethanol stimulation. So with that, I would like to acknowledge all people who have contributed to this work. As I have mentioned, Preben and Finn, unfortunately I don't have his photo, they have developed the quenching method or quenching of small amplitude limit cycle oscillations, which I have then used to characterize dynamic behavior in the Briggs-Rauscher reaction and extend the model, the mechanistic model of the Briggs-Rauscher system. And the work on the HPA axis has been done in collaboration with my colleagues from University of Belgrade, Željko Čupić, and unfortunately late Professor Ljiljana Kolaranić. And the work on the kindled animal model has been done in collaboration with Osama Abul Saud, who is now at the Mayo Clinic in Arizona. With that, I would like to acknowledge people in my research group. I am not actively working with them on this aspect. We are more working on developing experimental techniques and studying ethanol effects on the opioid receptor. And I thank you for your attention and be happy to take questions. Thank you.
Thank you so much for a great presentation. Do we have, what questions do we have?
I have a few questions. Yeah, I thank you for a very nice talk. It's very exciting to learn about this stuff. My name is Chiang. I'm from Emory University. Actually, by training, I'm kind of more a neuroendocrinologist, although I'm doing right now. So I have a couple of questions about your HPA model. First of all, does the ultradium frequency actually changes during the day? Like say for menstrual cycle or like say estrous cycle for the LH pulses, they tend to the frequency tend to change from depending on what stage the animals or the women are actually with respect to their menstrual cycle. So sometimes they kind of accelerate and say every 2 pulse hour, and then at some other time and it slows down to 2 pulses, 1 pulses per 2 hour. So in the HPA axis for the ultradium frequency pulses, they all the way the same. I see you have amplitude changes, right? But do you have frequency changes there?
So in an unperturbed HPA axis during a day, we have not observed in the model changes in the frequency of ultradian oscillations, or not really very big changes. But what is really good is that one can, so you have mentioned that sex hormones, for example, can make these changes. So one can couple the model to such changes and then simulate this behavior.
Okay, thank you. Yeah, my second question is for a supercritical Hopf bifurcation like this through negative feedback. Usually you need some nonlinearity or what we call ultrasensitivity to amplify the signal somewhere in your model, right? So in your network model there, I'm just wondering where that's nonlinearity coming from that's make the system oscillate. I took a snapshot of your equations. I'm just looking at there to see which part actually causing, providing this percentage amplification.
Sorry, now I went too fast. So you can see here, here we are having actually the feedback loops.
Oh, I see that. You have a square term there.
Sorry, yes.
I see. Okay. I think that in the previous slides, you show the ODEs there. I see there. Oh, I see. So for the ACTH feedback from cortisol, there, right?
Exactly.
Yeah, okay. And then there's some
Yes.
other places as well.
Yeah.
All right. Yeah. Thank you.
Yes.
Yeah.
Welcome.
So I would like just to go back to this quenching method. I was always impressed with the potential that it has inside. So I think that in exploring reaction networks, quenching method has to be equally powerful as perhaps nuclear magnetic resonance in medicine, or X-ray diffraction in crystallography or something like this. So do you have any idea why there are no more applications, why there are no more trials to apply this method in research?
I mean, I don't know, right? That's the short answer to it. As I have mentioned, the method has been developed some 30 years ago, and there is a number of publications. I think there are some 30, 50 publications where it has been used. What I could see is that it has a little bit created an interest, but I think, as in all fields, if one is not actively promoting something, then the number of people that applied is not increasing as rapidly. I mean, I can return the question to stoichiometric network analysis, which is also a technique that exists since several decades back, but it is not really renowned, even though it has huge potential. What is extremely important to underline, and there I agree with you really very much, typically when we have a complex oscillatory reaction, whether it is a chemical reaction or a biochemical reaction, we don't have so many species that we can monitor simultaneously. And even some species that we can monitor, are complex. So for example, if we use potentiometric measurements with platinum electrodes, then we see a very complex, actually we are monitoring a very complex phenomenon. So what is really good about the quenching method is that it gives quenching concentration and quenching phase angle, which are unique for each species and tightly coupled to the chemistry of the system. So yes, I think we should promote it a little bit more.
Yeah.
Thank you.
I agree with you. Thank you.
Well, I mean, if there are no further questions, maybe we can conclude, and everyone interested could just send an email and ask. We'll be happy to.
Absolutely. Thank you so much again, Vladana, for your amazing presentation. Really appreciate it. And sorry to keep you up late.
Thank you for the opportunity. It was really nice. Appreciate it. Thank you.
Thank you. Have a great day, everybody. Have a great evening.
Yeah. Thank you. Bye-bye.
Bye.